Hello and welcome to the Prelab video for Aspirin Part 1 Synthesis. This is an experiment in the General Chemistry 2 curriculum at York College of Pennsylvania. The goal of the aspirin experiment, which is Part 1 today of a 3-part experimental sequence, is to make a sample of aspirin and then to recrystallize a portion of that sample. We'll talk about what that means in just a couple of minutes. Aspirin's formal name is acetylsalicylic acid, pictured here on the board. These are line structures, which you may or may not be familiar with. At the intersection of any two lines would be a carbon atom. So this, at the top of this structure, for example, is a carbon atom having 3 bonds coming out of it, one to another carbon on the left, 2 to the carbon on the right. So the 4th bond, it's assumed when we draw line structures, would be to a hydrogen that is up on the structure here. So in reality, if we filled this out to all the other hydrogens that are present, okay, this compound would have the formula C7H6O3. Okay, so you're going to see these line structures in quite a few places moving forward in your chemistry curriculum. So it's best to adapt to them now, get used to them now. Anyway, aspirin, or acetylsalicylic acid, is an ester, a class of compounds in chemistry that feature this group of a carbon double bonded to an oxygen, okay, and then a single bond to an oxygen, and then it continues on either side of that. And so we have a couple of esters on the board here. That's one. This compound would actually be a double ester. It has 2 esters there, the acetic anhydride. And the process of making aspirin is what's called an esterification reaction. In it, we react a carboxylic acid, and a carboxylic acid is another functional group. It terminates a molecule, so it has carbon double bonded to oxygen and then an OH group attached to it, and then it would go off to some other component of the molecule. So we react carboxylic or an acid, okay, and turning it into an ester. Although here we're actually turning the OH into the ester in this case. So in this reaction, we begin with a carboxylic acid, salicylic acid. We basically cleave this molecule here and attach one of these pieces. So imagine this O now is what's attached to this carbon, and you arrive at the salicylic acid. The other portion of the molecule then would be free to bond to the OH that we have broken, cleaved there, and that would form our second product, acetic acid. So in this reaction, we have 3 acids present as far as we're concerned. Salicylic acid, and I've circled the carboxylic acid group on it. The acetylsalicylic acid, which has the exact same carboxylic acid group, and acetic acid. And so those 3 circled regions are actually the answer to Prelab question 3, which asks you to copy out this reaction into your lab manual and to circle the acidic part of each of the 3 acidic species that we have. So to conduct this reaction, it's pretty straightforward. We'll weigh out salicylic acid, which is a solid. Acetic anhydride will be a liquid. And then we mix these in the presence of a strong acid. We're going to use sulfuric acid. So we denote that in organic reactions simply with H+. Could be any of a variety of acids that help us accomplish this. We're going to use, as I said, H2SO4. Pretty concentrated, and that is an inhalation hazard. You don't want to be breathing in strong whiffs of the H2SO4 vapors. It can be very unsettling to breathe that into your lungs and cause you some distress. We'll be working in the hoods today, making sure you have this all set up in the chemical fume hood. This whole process is optimized if we conduct it at 80 degrees Celsius. And so you will immerse your reaction, which can simply take place in a Erlenmeyer flask, in a beaker. And you can then fill the beaker with some water that is at 80 degrees Celsius. And this will maintain your reaction inside the flask at 80 degrees Celsius. If we go lower than this, the reaction still occurs, but it occurs more slowly based on what we know about kinetics and temperature increasing the rate of a reaction. And if we go above this temperature, we actually get into a danger zone for decomposing some of our materials that we have, and then we wouldn't get the esterification reaction. When you're done, your reaction will have a solid inside the container, the Erlenmeyer flask that you've used. And so you'll want to filter out that solid acetylsalicylic acid from the other contents, the acetic acid, any residual acetic anhydride, and sulfuric acid that are left over. And so we'll use a simple filtration apparatus. Let me wheel one in here. Please remember, when you're setting up a filtration apparatus, a couple of safety points to keep in mind. First, you're going to do this in the hood because our vacuum apparati are set up in the hoods. At the back of the hood is where you find the vacuum line controls. I've used a ring stand to clamp my filter flask, which is the type of flask with the side arm on it, to the ring stand. This keeps the flask nice and stable. If we didn't have that clamped tightly, it would definitely be susceptible to falling over while we're working with it. So quickly clamp it into a ring stand. We use a Buchner funnel. That's one of the porcelain funnels with many holes in the base. And then a filter vac, this little black rubber ring that gives us a seal between the two things, the filter flask and the funnel itself. So this is your filter setup. You'll put a piece of filter paper inside here and then pour the contents of your reaction mixture through the filter. And allow them to filter out the liquid, which will be collected down here. And we're interested in the solid. We want to keep that solid. Now the solid product from the reaction will actually be a mixture of acetylsalicylic acid and potentially any unreacted salicylic acid. We may not have 100% conversion, in other words, of salicylic to acetylsalicylic acid. And so the follow-up weeks of lab are going to be designed to see how much salicylic acid remains in the sample, in what we call the raw sample of our acetylsalicylic acid. And to compare that to a really good sample, we are going to conduct a recrystallization. In a recrystallization, you basically dissolve your raw sample into a small volume of liquid, and you slowly cool the sample. So you would dissolve it into hot liquid, and then you slowly cool the sample. By doing this, you will form nice crystals of your product, acetylsalicylic acid. Now the reason this purifies our sample, okay, it's going to remove the impurity, which is salicylic acid in this case, is because the salicylic acid is less soluble in water than the acetylsalicylic acid, aspirin, is less soluble in water than salicylic acid. meaning it will be the first material to crystallize out, and the salicylic acid will stay dissolved in the water while we're getting the crystals of salicylic acid. And then we can filter off the liquid from our solid crystals of aspirin, and that should remove any remaining traces of salicylic acid that are present. Okay? So you'll be using the filtration device twice today. Once to get your crude products or your raw sample of products separated from the leftover liquids in the reaction, and then a second time to separate your pure crystals now of aspirin from the other material, potentially salicylic acid, that might be left over. You'll then store your two samples. So at the end of lab, you'll have your raw or your crude products, or raw aspirin, and you'll have your recrystallized aspirin. Place each of these into a separate container labeled with your name, ideally your lab section as well, so that they're easy to find when you come back to the next lab. Make some note, physical note of the properties of these, what their color is, the crystal structure. Is it a very fine needle-like structure or a fine powder, for example? These are physical properties that you can use to support your claim that you have aspirin. So that's getting a little bit ahead of myself, but in the three experiment series that we're going to do, your ultimate goal is to make this sample of aspirin, purify a portion of it through recrystallization, and then in the next 2 weeks to compare your pure sample to the raw sample, and also to some other commercially available samples of aspirin, to see how what you made in the lab stacks up against what you can buy in the store. So we'll do a series of qualitative tests in week 2, and then we'll do a quantitative test of the purity in week 3. Okay? You'll write one large lab report on 3 weeks worth of experiments, and so you need to take good notes during week 1 about what you did and what you observed, so that 3 weeks from now, when you go to write your report, you can recall what happened and have evidence to explain whether you have a good sample of aspirin or a poor sample of aspirin as a result of your experiment today. And that starts by taking good data, as I said, in the first experiment, so that you can reflect on that later on. Let's look at some of the other questions relating to the pre-lab for this week. They focus on the notion of esters, what these compounds are that we call esters. And so you're asked to consult chapter 20 of your textbook, which is where we cover some organic chemistry material. And in chapter 20, you can find examples of carboxylic acids and esters. Okay. A carboxylic acid would be any compound that has the COOH group terminating it. And so there could be any variety of carboxylic acids present. They may be drawn in line form or actually structural form, as I'm doing here. This carboxylic acid has 1, 2, 3, 4 carbons in it, so it would be called butanoic acid. Okay. An ester would be a compound that it has a similar component to the carboxylic acid group. It has the C double bond O and an O, but instead of terminating in an H, it goes on to some other part of the molecule. So for example, we could have a methyl group on one side and then 3 carbons on the other side, 1, 2, 3. So we would call this ester methyl, which is inorganic, meaning 1 carbon, and then propyl ester. Or another technique for naming these would be called this methyl propanoate. The OAte ending is often used when naming esters. Now, you may not, repeat, you may not use these 2 as your answers to the pre-lab question this week. Please look in chapter 20 and find 2 more examples, one of a carboxylic acid and one of an ester. It should be pretty easy, and the more of these you see, the better off you'll be in the long run. It should be pretty quick and easy to find 2 more examples in chapter 20. Okay. That was question number 1. Question 2 asks about 2 specific acids. It says, what acid is responsible for bee stings and bites? So bee stings and bites have methanoic acid in it. And this comes from the text as well. Okay. So methanoic acid would be just 1 carbon, and it would terminate with an H over here. Okay? This is methanoic acid. Again, meth meaning 1 carbon. All right? And this is sometimes called formic acid when we have just 1 carbon there. Okay. Pineapples have an ester in them. Many esters have pleasant smells associated with them. That happens to be one of their signature characteristics. You'll find a lot of esters in a variety of flowers and fruits and other good smelling foods. All right? And so in pineapple, the ester that is present is called ethyl butanoate. So following the pattern that we had over there, ethyl would be 2 carbons, so 1, 2, that would attach to our oxygen. And then the butanoate, this is our ester group, so that's carbon 1, 2, 3, 4, in line form. And again, these would be carbons filled out with any hydrogens necessary to bring each carbon up to 4 bonds. So that would be our ethyl butanoate. Right? So that covers the 3 pre-lab questions that you need to complete for this week. I believe we've gone through everything that's necessary for discussion in the experimental procedure. Again, be sure to work in the fume hoods with your reactions set up. And for your filtration, you'll have to do that to have access to the vacuum systems. Careful handling the concentrated sulfuric acid. Gloves are probably a good idea during that portion of the experiment. And if you have any doubts about or concerns about handling any of the chemicals, please consult with your instructor and the student TAs who are present in the lab who can help you and assist you with safe handling of any of the chemicals that you have today. All right? With that, good luck making your sample of aspirin. Really do a nice job in this experiment, because the sample that you make today or in this experiment has to last you for 3 weeks, 2 additional weeks of work. And if you were to rush through this experiment in the interest of getting done early, you'd be trading for a very low quality product today, I guarantee it, that you would then have to work with for the next 2 weeks. So it's definitely worth spending a little bit of extra time today to make sure everything gets done right and that you do a good job with this synthesis so that you have good materials to work with for the next 2 experiments. Good luck.